Okay, in this picture, you can see the ear on section. Just to quickly review the outer parts of the ear, you can see the auricle, also called the pinna. That's what's on the outside of your head, of course, that collects sound waves. In your lecture, we're talking a lot about this. If you've already had your lecture, you should remember some of these details of the outer part of the ear. All right, the auricle collects the sound waves and then channels those through this ear canal that's also known as the external auditory meatus, or it can be called the external acoustic meatus. But the vibrations, the sound waves then will hit the structure known commonly as the eardrum, but that's also called the tympanic membrane. All right, so right here I'm drawing a circle around the tympanic membrane. Of course, it starts to vibrate in response to the hitting action of the sound waves, and when it does, that sets up a bridge of bones to move. And those bones right here collectively are called the auditory ossicles, but individually they are the malleus, the incus, and the stapes. And as that bridge moves, it pushes against a membranous structure in the wall of the inner ear. All right, here's the inner ear. The outer part of it is mostly bone, except for where the little stapes connect. All right, so I'm drawing an area around the inner ear. And right here where the stapes connects, there's a membrane called the oval window. You're learning that for lecture. Don't worry about the name of that for lab. But anyway, the oval window gets pressed upon by the action of the stapes, and that sets up a fluid movement inside the inner ear. And that fluid is then starts a current that goes all the way around in this spiraling part of the inner ear known as the cochlea. In just a few minutes, we're going to see the cochlea on section, and we'll spend a good bit of time looking at that for lab. You'll be looking at some microscope slides of the cochlea. But just real quickly, let me go through the rest of the inner ear with you. This middle part of the inner ear is known as the vestibule, and connected to the other end of the vestibule would be the three semicircular canals. The vestibule and the semicircular canals both contain receptors for balance or equilibrium. The receptors for hearing are located in the cochlea, which we'll look at in a moment. If I backtrack to the middle ear and look, here's the middle ear cavity that the bones are in, but there's also a tube that leads away from the middle ear to the nasopharynx. That tube right here that I'm putting arrows on, that's called the eustachian tube. The function of it is to equalize the air pressure in the middle ear cavity with the air, here's the middle ear cavity, with the air pressure of the outer ear. And if those air pressures are equal, then that means the tympanic membrane is as flexible as possible. So air is constantly rushing up and down the eustachian tube, also called the auditory tube. But the air is rushing up and down that to make sure that the air pressure in the middle ear cavity is equal to that of the outer ear. When you hear your ears pop, that's when the air is rushing fast, very quickly, like when you're moving to altitude changes, such as when you're in an airplane. Or if you're diving deep in the water in a swimming pool or in a pond, you may hear your ears pop. That's because the water pressure in the outer ear is building up, and in turn, air will rush into the inner ear to try to compensate for that. Or excuse me, air will rush into the middle ear. Okay, I'm sidetracking a little bit. I apologize for that. Listen to your lecture recording for those of you who are in the lecture, and you'll get a better description of all of it. But right now, I want us to focus on the inner ear, and let's look more closely at the inner ear. All right, so if we move to the next slide, on this view, you can see the inner ear. And if you look at this section, there you see the cochlea. You can see it's somewhat like a tube within a tube. The inner tube is filled with endolymph. The outer, surrounding that inner tube, you have perilymph. If you look at this plane that sections it. Let's see if I can pinpoint that for you. Look at this plane right here that's sectioning it. Then if you look down at this bottom view, you'll see it on section. And you can see that in the cochlea, you've got just a spiraling of this tube within a tube pattern. All right, well, as the fluid starts moving in response to the movement of the oval window, as the stapes pushes against it, then that's going to set up a lot of fluid movement in this cochlea. And ultimately, that fluid movement is going to cause the receptor cells, cilia, to bend. So we'll be looking at that in just some of these future slides. Okay, now on the next slide that you see, you're going to look at a micrograph of the cochlea. Here, in this view on the bottom in the square, it's turned sideways, but on this next slide, you'll see it upright. And this is as if you were looking through the microscope. We'll wait just a moment for that next slide to come up. Well, let me click it again and see if it will. There we go. There we go. All right, here you're seeing a section of the cochlea. You can't see all of it. It's pretty large, even with the scanning power on the microscope. But if you look at, let's say, this circular area that I'm trying to identify right here. All right. Do you notice that you have 1, 2, 3 compartments? Okay, and so that would be like if you could uncoil the cochlea, just totally uncoil it and slice it on cross-section, that's what you would see. You can see that same thing if you look on the opposite side, like right here where I'm circling. Again, you're going to see 1, 2, 3 compartments. Okay, so if I clear this slide of the arrows and the drawings, all right, let's look at it a little more closely. All right, in just one of these circular areas with the 3 compartments, the upper compartment has a name. That's called the scala vestibuli, and it's filled with perilymph. The lower of the 3 compartments, also filled with perilymph, is called the scala tympani. And then the middle compartment, known as the cochlear duct, is filled with endolymph. Well, what happens, let's clear these arrows, what happens as the fluid in the scala vestibuli, that's the upper compartment, as that fluid starts moving, that causes this little membrane right here to vibrate. That membrane is called the vestibular membrane. And as that membrane vibrates, the endolymph here in the cochlear duct will start moving, which in turn causes this lower membrane to vibrate at the cochlear duct. That membrane right here is called the basilar membrane. All right, now sitting on top of the basilar membrane is the organ of Corti, and that's where the hearing receptor cells are located. If you look at some of these other views, let's say this one, and try to identify the compartments again, scala vestibuli, cochlear duct, and then in between the two is the vestibular membrane. All right, and then at the bottom of the cochlear duct is the basilar membrane, and sitting on the basilar membrane is the organ of Corti. Your book may describe the organ of Corti as also the spiral organ, because it literally does spiral around the cochlea as that basilar membrane spirals around. All right, now I want you to notice another membrane that's sitting within the cochlear duct. On these slide preparations, a lot of times it gets pushed up a little bit, but it's this little membrane right there. On one of the other views, look to the left, watch where the arrow comes up. There you see it again. All right, that's called the tectorial membrane. And in a fresh cochlea inside a person's ear, that does not ever move. So what's going to happen is, if I erase this, as the basilar membrane, right here, as the basilar membrane vibrates, this organ of Corti is going to be pushed up against the tectorial membrane, which is hard to see on this view because the tectorial membrane sticks up. I'm fixing to show you um, a more magnified view of this. We're going to look through just one of these sections. Let's look on the next slide. Actually, I believe this next slide shows this same cochlea farther down. I couldn't get it all in one field of view, so be patient and we'll see it move to the next. The computer's slow today. It's not moving from one slide to the next very quickly. Here we go. There you see a lower view of the same slide. For example, you can see right here, I'm circling on the lower right. All right, you can see the three compartments: scala vestibuli, cochlear duct, scala tympani. This would be right there, the vestibular membrane. There's your basilar membrane. You don't see much on the organ of Corti there, but you do see the tectorial membrane sticking out into the cochlear duct. All right, the next slide shows all this a good bit more magnified. All right, this is from another slide, but it's still showing this section of the cochlea. And right here, you can actually see the organ of Corti real well. The tectorial membrane is right there where I put the arrow. Let me clear all the arrows except for, let's put one on the tectorial membrane. On this one, you can actually see it goes down and is touching the top of the organ of Corti. All right, evidently the organ of Corti has moved up because of the vibration of the basilar membrane. And as it pushes up, tiny little hair cells or cilia are literally bent because they push up against the tectorial membrane. And if they bend enough, that's going to trigger an action potential to occur and to go all along the cochlear portion of the vestibulocochlear nerve, which is going to then go to the hearing centers of your brain. Okay, now I know I've spent a lot of time looking at all these different structures, but I find students get pretty confused on the cochlea. As far as your lab is concerned, make sure you know these parts. Make sure you know the scala vestibuli, vestibular membrane, cochlear duct, basilar membrane, tectorial membrane, organ of Corti, and then the lower compartment, scala tympani. Also, make sure you know what fluid is in the scala tympani and the scala vestibuli. That would be perilymph. And then in the cochlear duct, the fluid is called endolymph. So for lab, you'll just need to name all these structures and the fluids. You can see some good pictures in your textbook, also in your lab manual. If you'll look there in your exercise on the ear, you'll see these and you'll be able to label that. All right, I believe that's all for this section. So just make sure you know this slide on the cochlea real well and can point out these various structures. Also, for the model of the ear and pictures of the entire ear, make sure you know the structures that I've listed in your to-do list. Archive recording.